it is very huge developing field of interest, and particularly the interactions within the microbiota and also the metabolome, because we discussed the urine metabolites, and one of the colleagues from the chemical school, chemical university, they also have the project with some testing from the urine, the metabolites.
With metabolomics is really open field at the same.
Yeah, very
large.
Very promising.
Complicated.
Dámy a pánové, občerstvili jsme mikrobiotu, tak já bych opět podpořil naši osu střevo-mozek a je mi milou ctí, že mohu uvést druhou část dnešního semináře a vítám zde mezi námi pana doktora Vannucciho, který by přednesl své sdělení na téma mikrobiom a kolorektální karcinom. Někteří pacienti už se nás dneska ptají, zda třeba test na okultní krvácení do stolice bude nahrazen testováním orálního mikrobiomu, tak zatím říkáme, že ne, ale je pravděpodobné, že asi ano, tak je to takový úvod do přednášky. A ještě předtím, než předám slovo, tak bych skutečně rád upozornil na to, že Česká mikrobiomová společnost se nyní nachází na rozcestí a pokud společnost chcete podpořit, tak není to určitě takový nábor velký a reklamní okénko, ale můžeme teďka všichni rozhodnout o osudu České mikrobiomové společnosti, takže neváhejte stát se členy a také se aktivně podílet na problematice mikrobiomu a především také osvětě, kterou se snažíme šířit a navazování mostů mezi různými obory a také samozřejmě mezi uměleckými směry. Tak to jenom na okraj, předávám slovo a vítám vás zde, pane doktore.
Moc děkuji. Tady nemluvím česky, mluvím anglicky, protože bude moc lepší pro vás. In this half an hour of the presentation, I will do some kind of running around the question of the bowel, the colorectal carcinoma, how the interaction of bacteria can be, and how we have a demonstration practical in experiments that were done in my laboratory and demonstrate the difference between animals with and without bacteria. So, as told before, the microbiome is something that is a part of our body, of our essence of elements in the nature, and it is commanding in some way also our functions. And it is present in all organs that are on the surface of all organs that are in contact practically with the environment, internal and external. Now, these bacteria, but not only bacteria, because there are the viruses, because there are the fungi and so on, but we are focused on the bacteria. They can interact in the different places where they can either overgrowth or become assumed a pathological aspect. And it is known that around 15% of all cancers that develop can be correlated with bacterial function. And that are known promoters of cancers, bacteria like Helicobacter pylori for gastric cancer, Salmonella enterica serovartifi for gallbladder cancer, Streptococcus bovis for colorectal cancer, and we will see that for colorectal cancer there are much more than this one. Certain type of Escherichia coli, still for colorectal cancer, but related to inflammatory conditions like Crohn's or colitis. Chlamydia, chlamydophilia pneumoniae for lung cancer, and oral streptococci for oral cancers. But these are some of the correlations. Now, we try to focus on the bowel, And we know that the relationship between bowel and bacteria is something that is creating a universe that we are developing to know continuously. Because there is the bad and the good, there is the known and still a lot of unknown. And the bowel, the colon, it is a fantastic organ. A fantastic organ also for the scientist, because not only is providing the final digestion of our food, of all we introduce, or recovery of water and many other things, but it is an immunological reserve, because there is the widest immunological organ, the lymphatic organ, that is present in the body, an organ that can condition also the function of the rest of the body, immunological and not. And because the presence of continuous pressure by the bacteria, it is a place where the fight is controlled to don't become dangerous, destructive. So there is an activation without inflammation, or we can consider a tolerated level of inflammatory condition that maintain the bacteria in their place, co-working with us, and in the same time maintaining active the immune system to be ready eventually against the pathological ones. So it's a fantastic harmony of system. And it's great, this one, because it's permitting to have a living place, a living laboratory in which we see these effects. All these one pass through relationship between antigens that can be processed by the colon mucosa. If this one is working properly, they are then elaborated by the dendritic cells in the direction of tolerance, production of local antibodies like IgA, but on the other side also cells, immune cells that can tolerate and diffuse the tolerance toward this one. If they are not passing correctly, and they are just crossing the mucosa, they are doing regularly what is happening in every other part of our body, so stimulating the inflammatory process, acute, and by this way, creating all the problems that we have. So, a point that is important, we need to have not only a good immunological defense, but also a good barrier, because this is one of the first defenses against the bacteria and the right processing of the antigens that are. Then it's not finished. We know that we are speaking now about tumor microenvironment or general microenvironment. This is a very good concept because it's giving the complexity of our body in every part of our body. And here is an imaging that can collect altogether different elements that are cells, nerves, fibers, production of metabolites, elaborates, etc., that altogether every microseconds are working in our body, changing in our body, giving in every place of our body the life and the modifications and the regulations to stay alive here in the world and inside with all the things that we have to maintain. And if these processes are going not well and we have cells that are going to be transformed, creating clones that then activate danger signals and process for creating a tumor. If the process is taken in the beginning by a right inflammatory reaction and the development of adaptive immunity, completing the work of the innate immunity against the cells. If it is not going, a chronic inflammation and altered inflammation can be grow up and maintained in the place. Now, chronic inflammation is something important. We have to know that there are, for example, some bacteria has not the same kind of relationship with the mucosa. So there are some, here is a nice paper, in which they were tested some bacteria. So it was seen that some were able to stay and not modify the mucosa. Others were maybe enforcing even the resistance of the relationship between the mucosal cells. Others, they were opening the way within the mucosa for the access of either bacteria or materials from bacteria that can activate inflammation. Procarcinogenic bacteria in the colon, there are various, putative or more directly demonstrated. Here we see some kind of Escherichia coli, the Bacteroides fragilis, Campylobacter jejunum, that is working, we know, in the stomach, but is able eventually to translate and arrive to be found even in the colon, in the cancer of colon cancer. Then, here there are others, but in the top, you see the Fusobacterium nucleatum. Now it is the star of the bacteria. It is in the top 10, the winner of the Cancer Cup. And it is considered one of the most relevant for directly function because it is acting on double-strand DNA, creating breaks and mutations. On the other side, because it is quite constant, there are at least 70% cases in which you can see the presence of this bacterium associated to the cancer. It is a bacterium that it is present normally in the mouth, in the oral cavity, but is resistant to the acidity of the stomach, can transfer in the bowel, and at the end can become a symbol, a sentinel, also the presence of this one. Moreover, because it has the possibility to be adhesive, looks that is able to transfer together with metastatic cells, and it is found also in metastatic places. So it is very interesting. It is considered a possible marker to be studied, to be used for seeing if people develop cancer or has a suspect of cancer. Surely something that must be studied because these peculiarities and promising effect, because if it's sticking with the tumor, it can be used also as a target for bringing there something that is catching this bacterium, bringing together a chemotherapy or other things, and killing the tumor, or making evident the tumor. So doing some theranostic approach, just focusing on a contaminating bacterium. So it is another step how to look of bacterial microbiome. Then, the tumor in, the colorectal carcinoma has an evolution that is long in the time, and it is by different steps of mutations that are happening. Now, the steps of mutation are related also from the local conditions. So, from environmental factors like what we eat, what we breathe, what we are drinking and so on, but also what is the environment that is in the bowel and the type of bacteria that can be there. And for this one are useful, for example, studies that are in conventional animals, so with the full microbiome, or germ-free animals that are completely sterile, inside and outside. So they are a plate where we can do our experiments in the case. But another thing is that it is found that the bacteria can resident also in the cancer. I mean, can contaminate and maybe can drive the evolution. Some bacteria may be positively controlling, some bacteria instead fueling, stimulating to grow further. It is true that the mucosa in that place is not normal and can, in this way, bring the possibility to stimulate also the local inflammation. This is what is appearing, the bacteria in the mucus of a rat, conventional, you see, and they die. And this one instead is the clean mucosa of the germ-free animals. Now, the germ-free animals have various characteristics that demonstrate something that is some way softer, relaxed in the tissues, especially in the gastrointestinal tract. And these are very well visible here. This is a normal cecum, and this is the enormous, really softener cecum of a germ-free rat. And this one in the mouse. We have the same. Before, it was indicated some relationship with the neurosystem and the possibility of influences. And this is important because all the stimulation immunological that is present, activated by the bacteria, can have effect also in the transmission of signals, the tone of the muscles, and the relative capacity to have normal function, also motorial in the bowel. Moreover, the immune system is not less functioning, is working the same, but is less developed, less evolved than in the conventional animals. Here there is a very small follicle in germ-free, and this is how it appears in the conventional animals. If we put the bacteria, if we take the germ-free animal and we put in normal condition, we see that from this, in a few days, is becoming like the regular one. Is arriving tone, is activating everything. But why? Because it's activating the immunity there. It's activating all the functions that are present. Look, that is true because this was the original follicle. And this is after 4 days. We see evolutionary adaptation important because there is adaptation that we demonstrate of the mucosa structure. We have to consider that all these bioses, they can modify not only the surface of the bowel, but the same structure of the bowel. And by this way can affect in some way even the responses inside. This is a human one. We see very nice structure. We see when near a tumor, and we see the cells, the dots, the white dots that you see, they are inflammatory cells on the border of tumor. And here we see inside the tumor that is completely gone, the regular structure, and we have the inflammatory cells that are present there. Now, these inflammatory cells, they have to fight directly with bacteria that can go and stay inside the tumor. I repeat, can be bacteria that have a positive function in trying to slow down the capacity of the tumor and to increase the response of the immunity in the place. Others that instead are completely from the side of the enemy. And bacteria be dangerous, for example, in chemotherapies, because they can sometimes modify the structure of therapies that are given. This is the case, for example, a study on the pancreatic cancer, gemcitabine, and was seen that finding bacteria present even in the tumor in the pancreas, etc., they were able to split, to modify the molecule, to give them inactivity toward the tumor. On the other side, they can be good and bad bacteria that can cooperate with the immunotherapies. And you know how immunotherapies have become important in our years recently. Finally, we have to consider that using probiotics, we can introduce bacteria that can save the place by the production of short-chain fatty acids and the butyrate that is making better conditions in the place. But if we are looking in the literature, we find that they can be used, the bacteria, the intratumoral bacteria, as a target for therapies, as I told, directly against the tumor, using like a Troy horse. That can be for precision medicine and in immunotherapy modifications to control this one for efficacy. That can be the bacteria used if they are attaching to tumors or they are entering, like Salmonella, in cells to be used like therapy, to modify and to become toxic, bringing something inside or directly to the cancer cells. So there is a future for the bacteria. Now, in the last minutes, I will show you an experiment that we have done, controlling how to grow the tumors with the same model in germ-free and conventional animals. So this is the model is quite simple because there is the production of a hernia, this one you see, by putting the top of the cecum of the animal through the wall, below the skin. There is this bubble. We can inject what you want, going directly from the top, from the beginning of the large bowel, and by peristalsis is going inside. So we can regulate all our experiment. And we were doing this one in conventional and in germ-free animals. Now, this is another thing how to do these experiments is to associate or not inflammation, a colitis. If we are doing the simple experiment without inflammation, this is the trend of growth and development of tumors in the time. In 32 weeks, we have the full evolution. In 8 months, we arrive to have a metastasis. But if we are doing the inflammation, you see that there is an anticipation of at least 2 months on all the processes, the importance of inflammation. When we are going, what is happening, induction, we see that the tumor in yellow are the ones from the germ-free animals. No bacteria in the bowel. And you see that the violet, they are in the bowels with bacteria. They are much higher. Multiple, Generally, in the germ-free animals, a single or just a couple, smaller and instead more evoluted in the eye. It is the same when we are giving inflammation, it's the same. So, the difference are the bacteria. It means that the level of tolerance to the carcinogen and the events of carcinogenesis, it is lower in the conventional animals, so the animals that already have immunity that is activated, have a lot of background by this condition, and have a higher tolerance to many antigens in comparison to the germ-free that are much more naive. They are much prone to react because they have and to tolerate eventually events that are produced by the carcinogenic pro-inflammatory stimuli and so on. So this is an indication how bacteria can be important in the evolution of colorectal carcinoma and probably in other cancers, how modification, dysbiosis, can be important in modifying membrane, the mucosal surface, but even possibly the structure and surely the reactivity in the bowel, and how bacteria in their different species can be differently acting, positively, negatively, and some can be used eventually as a future target, either for diagnosis and for treatment. So I conclude because it's my time, and this is my laboratory. I have to thank all these people. Then there are others that were with me before, I'm not yet present here, that produced some of the data before. This is all the grants, etc., and collaborators that we had, and I thank you. Moc děkuju za pozornost.
Thank you very much for your